afternoon, students. Today we'll take up the study about the most interesting molecule in the living system, and that is the DNA. This DNA you already studied in your previous classes, so I just want to ask you, what is the full form of DNA? Please don't give collective answer one by one. Yes, Matt? Deoxyribonucleic acid. So those who don't know the spelling and other, they can revise it from here. So DNA means the deoxyribonucleic acid. Now in this letter there is a word acid. So that means this DNA is acidic in nature, right? So this molecule is acidic in nature. So anybody knows about the name of that scientist who first of all elucidated that DNA is acidic in nature? Anybody? No. So anybody? No. Friedrich Miescher. Yes. So it was Friedrich Miescher who first of all said in 1869 that DNA is acidic in nature, and he called DNA by a different name, and that name was, anybody knows the name? Yes, we are nuclein. So he called it nuclein. Fine. So in 1869, Friedrich Miescher said that an acidic substance is present in the nucleus, and he called it nuclein. Fine. So this was about the history of DNA. But you see, the double helical and the most interesting structure of DNA was given by Watson and Crick. For that they got the Nobel Prize. But that they elucidated in 1953. This model was proposed in 1953. Very long distance. Reason was because DNA is a very long molecule. So it was not possible for the scientists to separate that, the long molecule, and study its structure. So that's why it took a very long time. We'll study about its double helical structure later on. So let us again come back to the DNA. Now DNA, as you can see in this diagram also, it has made up of two chains of nucleotides. So since it is a polymer and it's a very long molecule, so these two chains are made up of small units of nucleotides. So can you imagine, can you guess as how many nucleotide units might be present in one DNA molecule? Any guess? कितने nucleotide units होंगे approximately? Millions or billions? It is approximately 6 billion. Right? So exact number is 6.6 into 10 to the power 9. Base pairs, mostly we say the nucleotides. So let us again come back to the single unit of nucleotide chain. So let us observe from this diagram. So as you can observe in this diagram, there are two chains running anti-parallel to each other. This is one chain and this is the another chain. They are helically coiled, and this structure was given by, as I said, Watson and Crick. For that we got the Nobel Prize in 1953. So Jameson, Watson and Francis Crick. But this experiment, this structure, double helical structure, which is mostly accepted by all the scientists all over the world and have got very important hallmarks in it. We'll discuss about that the number of adenine, the base pairs, they are equal to that of the thymine, and the number of guanines are equal to the cytosine. So this was a very important hallmark, and the base pairing, which explains all the complementarity and how the DNA replicates itself, how it transcribes itself. So that is all based on this important hallmark that adenine pairs up with thymine and guanine pairs up with cytosine. We'll discuss about this. But before that, I would like to explain the single unit of nucleotide. So this single unit of nucleotide is made up of three components. I just want to ask from you people that what are the names of those three components of which this single nucleotide is made up of. A single nucleotide तीन components को मिला के बनता है. So what are the names of those three components? Yes, Jyoti. Very good. So one is nitrogenous base, another is phosphate group, and the last one is the sugar, pentose sugar. pentose means the 5 carbon atoms are there. So just to explain you people, first of all the names. First was, as you said, nitrogenous base. Then it was pentose sugar, and the third one, phosphate group. So these three are the components which constitute one nucleotide, and then so many nucleotides are there, so it is a chain, polynucleotide chain of nucleotide. And there are two such chains in DNA, as you can observe, and those two chains are helically coiled. This structure was given by Watson and Crick. We call it very famous structure. We call it double helix. Double helix. Fine. So again we have to categorize the nitrogenous bases into 4 types. There are 4 types. 4 types of nitrogenous bases are there. I would like to know the names of those 4 nitrogenous bases from you. Yes, Tane?
Nitrogen bases are adenine, guanine, cytosine, thymine, and uracil.
Yes. So there are 5 nitrogenous bases. At a time 4 are present, and in case of DNA we don't have one base. And what is that one base which is not present in DNA? Uracil. So uracil is present in RNA, and rest 4 they are present in DNA. That is adenine, guanine, cytosine, and the thymine. So again there is a categorization between these 4 bases. Out of these 4 bases, a few are purines and the rest are pyrimidines. Fine. So what are the names of the purines? What nitrogenous bases comes under purines? Yes, group. Adenine and guanine are the purines. Very good. So adenine and the guanine, they are the purines. Fine. So the rest are the pyrimidines. What are the names of the rest of the nitrogenous bases? Uracil, thymine, and the cytosine. Very good. ये तीनों हो गए आपके बचे हुए pyrimidines. Fine. So pyrimidines and the purines. Very important feature of DNA is that when base pairing comes of the nitrogenous base pair, purine always comes opposite to pyrimidine. That is why the distance between these two segments, these two polynucleotide chains, is always constant. Purine always comes opposite to pyrimidine. So chemically the structure is such like the distance remains the same between the two polynucleotide chains. Also one plane stacks over the other, that is also gives additional stability, as well as the distance of the two chains remains the same. Fine. So let us come to another point then. You talked about the RNA and the uracil. So what is the full form of RNA? Because this is also a very important molecule. What is the full form here in group RNA?
Ribonucleic acid.
Ribonucleic acid. So the full form of RNA is ribonucleic acid. This is also a very important genetic material in viruses. In most of the viruses, RNA is the genetic material. So the difference between DNA and RNA is that lies in the sugar molecule. जैसे कि हमने अभी न्यूक्लियोटाइड का कंपोनेंट्स स्टडी किए, that nucleotide is made up of one sugar, one phosphate, and another nitrogen. So the uracil is present in RNA and thymine is present in DNA, but there is a difference in sugar molecule also. In case of DNA, this is a diagram of ribose sugar. So in ribose sugar at the second position, OH group is present. So this is the ribose sugar. Fine. And when O is not there, when O is removed, it is deoxy. So that deoxy sugar is present in DNA. So deoxyribose sugar is present in DNA and ribose sugar is present in RNA. So that's why I shown it like this, so that you can remember it. So this is the difference, that one is uracil, another is in the sugar molecule. Rest is one more very important difference between DNA and RNA. As you can observe in this model, so there are two chains running anti-parallel and they are helically coiled. But in case of RNA, how many chains are present? Only one chain is there. There are not two chains. There is only one chain, and that is also made up of nucleotides. So nucleotide chains are there. That is okay, but there is only one chain in RNA. That function we will study later on. Today we will take up the DNA. So now, as you can see, observe in this diagram of this DNA, that two DNAs are being formed from the parent DNA. These two are the daughter DNAs. So these daughter DNAs are exactly the replica of the parent. Also, they are the replica of each other, and they are the replica of the parents also. That means all are identical. The two daughter DNAs are identical to each other as well as to the parent one. What is the reason? The reason lies in the complementarity of the base pairs. You see, both the strands, both the polynucleotide strands, there are two strands. Both these strands can act as a template. Both the strands act as a template and both can replicate themselves. And for the replication, the scientists, Watson and Crick का वाला Crick said that there is a complementarity of the base pairs. जैसे हमने अभी डिस्कस किया, adenine always pairs up with thymine and guanine pairs up with cytosine. So at the time of polymerization, at the time of replication, so since they two are complementary to each other, so that the new strands are formed, which are also complementary of each other. As a result, the two daughter DNAs are exactly the replica of each other. So that is the reason. So so many concepts can be explained by means of the double helical structure. Now next we come to the length of the DNA. We say that DNA is a very long molecule. Initially I told you that is why it was delayed in its the proposition of its structure, because it is a very long molecule. So we just now discussed that there are about 6 billion nucleotide sequences are there, the units are there. Polynucleotide chain में 6 billion जो है वो nucleotides हैं. We instead of nucleotide we can also call base pairs. So there are exactly 6.6 into 10 to the power 9 base pairs. Now let us calculate the length of the DNA. Let's try to calculate. For that purpose, we should know the two facts and figures. But first is how many total nitrogen bases are there in total DNA. पूरे DNA में कितने nitrogen base pairs हैं? So that we know. Just I said 6 billion. So exactly is 6.6 into 10 to the power 9. One more thing we should know, what is the distance between the two nitrogenous base pairs. एक पेयर बेस पेयर ये है, दूसरा बेस पेयर ये है, तो दोनों के बीच में डिस्टेंस कितना है? If we know that distance, we can easily calculate the total length of the DNA. So I just want to know what is the distance between two nitrogenous base pair, one pair and the next pair. Yes, said nanometers. Units are very important. So 0.34 nanometers, that is the distance between the two nitrogenous base pair. One more additional question. This is actually a one turn, complete turn, which is in the right-handed fashion of DNA. So this one complete turn includes 10 base pairs. You can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is a single turn. एक turn में 10 nitrogen base pairs are there. And the distance between two nitrogen base pairs is 0.34 nanometer. So what is the total distance of the pitch? Yes.
2.2.
Only pitch. This much. Not the entire DNA. Only this 10 base pairs का कितना होगा? एक distance 0.34 है. 10 base pairs are there. Simply you have to multiply with 10. 0.34 you have to multiply with 10. What comes out? 3.4 nanometer. So 3.4 nanometer जहाँ ये distance दिखा रहे हैं. So this you can focus it here. So this complete turn has got 10 base pairs and the distance is 3.4 nanometer. Now let us calculate the total length of the DNA. So as I said, we have to multiply 6 billion base pairs with the distance between the two base pairs, and we just said it is 0.34. Now when we multiply करेंगे, it comes out to be 2.2 meters. So always remember it is approximately 2 meters. हमारा जो DNA है उसका पूरा लेंथ जो है 2.2 meters है. Now एक और बड़ी very speciality of the DNA. The 2 meters DNA has to be packed inside a space which is only 10 to the power minus 6. That is the size of the nucleus. We just said 10 to the power minus 6. So the size of the nucleus is 10 to the power minus 6. 10 to the power minus 6 डायमेंशन में 2 मीटर्स का डीएनए कैसे accommodate होगा? So that is a mirror. So that the special feature of the DNA is its coiling, its super coiling, and the very high level of coiling, and the proteins which are supporting this coil. So let us study the interesting feature of this DNA and the coiling of the DNA. As you can observe, this is a ball-like structure. So this is not a ball, actually it is reflecting, it is depicting a protein. Proteins are helping the DNA in coiling, especially because space is very less, DNA is very large, so it has to be coiled in a very high level of fashion, in a very good fashion. It has to be coiled. So that coiling is like this. Suppose that is concentrated on this diagram. Now, proteins, as I said, they are there. These are the special type of proteins which we call histones. The special type of proteins present in the nucleus. Now, proteins, how they are arranged? They are arranged in the form of units. They are arranged in the form of groups. In one unit, this diagram is of one unit which we call histone. This unit is made up of eight molecules of this protein. So 8 molecules of proteins together form a structure which is known as histone. Fine. So this is a histone. We call it histone octamer because 8 units are there. So we are calling it histone octamer. So this is the histone octamer. 8 molecules are there and they are constituting one unit. Fine. So millions of such type of structures are present in the chromatin fibers. When we observe this DNA or the chromosomes under electron microscope, we observe beads on string like of structure. Beads on string. ऐसे beads जैसे string क्यों होते हैं? This kind of structure is observed in the electron microscope. Those beads are nothing, but those are the nucleosomes. So we call this histone along with the DNA which is wrapped around it, histone octamer along with the DNA which is wrapped around it. Together we call it. What is the name given to this structure? Anyone? Yes, nucleosome. So this structure is known as nucleosome. So nucleosome means histone octamer and the DNA which is wrapped around it. Now I just tell you that there are 200 base pairs wrapped around one histone octamer. एक histone octamer के चारों तरफ कितने base pairs wrapped हैं? 200 base pairs are there. So you can calculate that how many nucleosomes will be there in your human genome because you know the length of human genome, you know the total nitrogen base pair, so you can easily calculate how many nucleosomes are there. This question I am going to give you the homework. ठीक है? तो ये आप करके लेके आएंगे calculate because you know every facts and figures. So this was about the nucleosome and the histone octamer. But this is said it is a very long molecule. So in addition to these special type of histones, the DNA also needs some non-histone protein molecules just to give it a support. The support के लिए some non-histone proteins are also there which are supporting this special level of packaging of DNA. Okay. Now let us come to the flow of information. Why we are calling it DNA is very important. DNA is very important. It is the most interesting molecule in this entire living world. Why we are calling it? Because it has got the genetic information. It can preserve that information throughout its life, throughout the life of that individual, and it pass on the same information to the next generation. That is why we say it is a very important molecule. So about this molecule, how the information flows from this DNA, how the information comes out from this DNA. Now I just want to ask you the name of that scientist who first of all given that how that information comes out from the DNA. What was the name of that scientist who has given the flow of the genetic information from DNA to outside to the cell? Yes.
Francis.
Yes. Francis Crick. He is the same Crick. Watson and Crick. Okay. So that Francis Crick has given the it told us that the information flows from DNA to RNA. DNA never comes out of the its nucleus. Even if it is Escherichia coli or the prokaryotes, prokaryotes mean the bacteria. So in bacteria, they don't have a proper nucleus. That is why we call them prokaryotes, because they don't have a well-defined nucleus. But even then, that DNA is not appositely placed there. It don't move from one place to don't roam inside the cytoplasm. It remains at a specified position only because of this special type of packing which we just now discussed. That proteins are there to assist. So proteins are there which can keep it in one place, and we call this a special place as nucleoid. This you have studied in lower class. The nucleoid is there. So nucleoid is a special place where DNA remains, even in the absence of the nuclear membrane. So this was about the DNA, and eukaryotes may have explained you that histones are there and the non-histones are there. One more important factor. Since the two are totally different, DNA and proteins, they don't have any similarity. They don't have any similarity. DNA, what is the single unit of protein? What is the single unit of protein? Yes, amino acids. And what is the single unit of DNA? Just now studied. Nucleotide. So DNA is made of nucleotides. Proteins are made up of amino acids. There is no similarity between amino acids and the nucleotide. But even then, the information is flowing from DNA to protein. Proteins are made as according to the information in the DNA. So this genetic information was first of all this flow of information, how it flows. It was given by Francis Crick, and we call it as, what is the name, special name given to this flow of information?
Central dogma.
Very good. Central dogma. So central dogma is the special information which flows from DNA to proteins via RNA. Now, I just want to know the names of very special types of phenomena, and I will ask you, name that process by means of which DNA gets replicated from DNA. Let's say यहाँ पे हो रहा है, we are getting two daughter DNAs from the parent DNA. What do you call this process? Yes, Priya? Replication. Very good. So this is replication, means DNA to DNA. This A was actually replication. Now DNA can replicate itself. Can protein replicate it themselves? Proteins cannot replicate. That is why proteins are not the storehouse of the genetic information, because they cannot replicate it themselves. DNA can replicate. Now, after replication, that's okay. From one DNA to DNA can be formed. Now, the flow of information is from DNA to messenger RNA. So, what special name has been given to this process? Yes, Manmeet?
Transcription.
Very good. This is transcription. So, transcription means the flow of this genetic information from DNA to messenger RNA. Fine. Now, the process is not stopped there, because this we have to form the synthesize the proteins also. So the information will flow from messenger RNA to proteins also. What do you call this process?
Translation.
Yes, Kismat? Translation. Very good. So this is translation from RNA to proteins. So this is the central dogma, that is the flow of information from DNA to messenger RNA and from messenger RNA to protein. Fine. So, and this central dogma, the concept of central dogma and the proof was given by Francis Crick. Fine. So, now, very interesting question. This information, can it move in the reverse direction also? That is, can RNA can give the information that RNA can produce DNA, or DNA always produces messenger RNA? Can this process goes in the reverse direction also? It is yes. Now, okay, very good. So, what do you call this reverse process? Yes. Very good. This process is known as reverse transcription, because this process from B to C, from DNA to RNA is transcription, and from again RNA to DNA it is reverse transcription. Now, where do you find this reverse transcription? In which molecule, in which organism you find this reverse transcription? HIV virus. Yes, it is there. And mostly viruses, some of the most viruses are not various types. But in HIV virus, this information flows in the backward direction. So there RNA can produce DNA. ठीक है? So this process is reverse transcription, but this is possible only due to the presence of a special type of enzyme. What is the name of that enzyme which facilitates reverse transcription? Yes, Sufi.
Reverse transcription.
Very good. Reverse transcriptase. ASC लगेगा उसके बाद. So reverse transcriptase is the enzyme which helps viruses for reverse transcription. So this is the information when it flows in the backward direction. Now I just not told you that adenine pairs are talking about the pairing. So let us try to make our own, replication, own polymerization if we know the sequence of base pairs in the template strand. So अगर template strand के nitrogen bases हमें पता है, so can we make it's on the basis of the complementarity of the base pair that adenine pairs up with, that we know it pairs up with thymine. So can we make a new strand? Can we? Yes. Okay. So let us try to make. If this is the template strand and the first nitrogen base is adenine there, what will be its complementarity on thymine? One by one. Yes, Anand. Adenine, thymine. Then thymine, adenine. Guanine. One by one. Yes. Guanine, cytosine. Cytosine, Ashu. Guanine. Adenine, thymine. Thymine, adenine, not adenosine. Guanine, cytosine. Cytosine, thymine. Likewise. So this is the way you can know, if you know the complementarity of the base pair, we can make our own. By just now we can make it a new a strand just on the basis of the complementarity of the base pair. This was a very important hallmark in double helical structure given by Waxman and Crick. Now there were two very important scientists who helped Waxman and Crick in elucidating, in proposing this important data, and I will tell you the names of the scientists. You have to tell me the word. The names of the scientists were Rosalind Franklin, Maurice Wilkins. Maurice Wilkins and Rosalind Franklin. These two scientists, they assist Waxman and Crick. In what way they assist? What was their work? Yes, Noor? Yes. X-ray, the model, the most important model which is accepted worldwide as the double helical structure of DNA, and this model was based on, this proposition was based on the work of Maurice Wilkins and the Rosalind Franklin, and the work was on X-ray diffraction method. So X-ray diffraction method, according to that, they discussed they proposed this model. So let us again discuss the salient features of this double helical model of DNA. So as you can observe in this diagram, in this model, there are two strands which are running and in anti-parallel direction. Why we say anti-parallel? Because both these strands have got the polarity. You can observe it. On one end it is 5 prime, and on the other end of the same strand it is written 3 prime. If 5 prime is on one end, 3 prime is on another end. So before discussing what is 5 prime, what is 3 prime, I would like to clarify this concept. Again come back to the sugar molecule, because sugar molecule is present in both DNA as well as RNA. In DNA there is deoxy, and in RNA it is ribose sugar, but pentose sugar is present in both the cases. So pentose is the 5 carbons. Let us count. How many carbons are there? 1, 2, 3, 4, and the 5th one. 5 carbons are there. So when 5th carbon end is there in any of the polynucleotide chain, if the free end is there, we call that end as the 5 prime. This 5 prime is nothing but is the 5th carbon. And if on the other end, this is the free part, 3rd. Because phosphate is attached with it, if the phosphate is not there, we call it this end as the 3 prime. So 3 prime is the phosphate molecule and the 5 prime is the 5th carbon atom. So that is the 5 prime and the 3 prime. So two ends are there, so polarity is there, so we call it we say that they run in the anti-parallel fashion, in anti-parallel direction. One chain is running from 5 to 3 in this direction, and another is chain running in the opposite direction. So उसका 5 and 3 का उसके opposite होगा. Find this point is clear? Okay. Now these two strands are, first of all, they are coiled helically in a right-handed fashion. Fine. We are discussing the salient feature. Now let us ask some questions and try to get the answer from the people. So as we have just now discussed, there are 4 types of nitrogen bases. I want to ask you, adenine, okay, it pairs up with thymine. So many times you have given me this answer. One now just observe this model very carefully and tell me how many bonds are present between adenine and the thymine. How many bonds are present? Observe it. Yes, Madhu. 2 bonds are present. Fine. So these 2 bonds which you are just observed, these 2 are the hydrogen bonds which are present between adenine and the thymine. Okay. Now observe the rest of the 2 pairs, guanine and the cytosine. How many bonds are present in guanine and cytosine? 3. So there are 3 hydrogen bonds. So this is fixed. Guanine pairs up with cytosine by means of 3 hydrogen bonds, and adenine pairs up with thymine by means of 2 hydrogen bonds. Now, 2 more important bonds which we have to study, we have to remember. Now, these 2 chains of the DNA, which we call the backbone of this DNA molecule. Okay? In this backbone, 2 important molecules which we discussed in the very beginning, nitrogen base, phosphate and the pentose sugar, they are arranged in an alternate fashion. Phosphate and the sugar, alternately they are arranged. It is on the backbone. And the nitrogen base is the project inwards. Okay? So now, what is the bond which is present between sugar and the nitrogen base? Because nitrogen base is present inside, sugar forms the backbone. There is a very special type of bond which is present between nitrogen sugar and the nitrogen sugar and the nitrogen base. What is the name of that bond? Anyone? Yes, Amanat? Glycosidic linkage we used to call it, and we the more specifically we go N-glycosidic linkage. That is the linkage between this sugar, which is this time it is deoxyribose sugar, and the nitrogen. So what is that bond? N-glycosidic linkage. Fine. One more bond left, because phosphate is also there. So phosphate is attached, is bonded with sugar molecule by means of a linkage. What is the name of that bond? Noor. Phosphoester. Phosphoester. Okay. Then 2 nucleotides are there. What do you call it? Phosphodiester. Phosphoester with 1 and 2 nucleotides are call it phosphodiester. Okay. So these are all the bonds. Now let us recapitulate some very important points. How many nitrogen bases are present in a human genome? How many nitrogen bases are present in human genome? Raise your hands. Total human genome. Yes, Vicky. 6.6 into 10 to the power 9 base pairs. Okay? What is the approximate length of human DNA? Riya. DNA का length 2.2 meters. 2.2 meters. Now note down the homework also, students. You have to calculate the number of base pairs in Escherichia coli. Calculate the number of base pairs in E. coli. Number 1. Then you have to write down the salient features of DNA, and the last one you have to draw the diagram of DNA showing the replication. Fine? Okay. Thank you.